Now, all human cells rely on the metabolism of your macronutrients, like we've already discussed, so your glucose, your fats, your proteins, in order to generate energy for survival and function. Now, each of these macronutrients feeds into specific chemical pathways. However, glucose is the body's primary and most immediate source of energy, particularly for tissues that have limited fuel flexibility. So, glucose or carbohydrates will be the primary fuel source out of all of your macronutrients. Now, the central nervous system, which is going to include the brain and the spinal cord, is highly dependent on glucose for its energy needs. And unlike skeletal muscle, which can utilize fat for fuel, the central nervous system can't always effectively metabolize fatty acids because they don't cross the blood-brain barrier easily. And this is why maintaining blood glucose levels within a narrow range is critical for brain function. So when blood glucose or blood sugar drops too low, cognitive and neural performance can decline rapidly. Now, most other body cells are metabolically adaptable, so they can switch between carbs and fats and sometimes, if necessary, proteins, depending on availability and energy demand. So, for example, resting muscle cells primarily burn fat, while working muscles rely more heavily on carbohydrate metabolism because glucose oxidation is faster and can support higher energy turnover. Now, when we talk about carbohydrate metabolism, we focus mainly on glucose because it's the most common and most efficient carbohydrate fuel in the body. And there's two main metabolic pathways that handle glucose. You have glycolysis, which occurs in the cytoplasm, breaking down glucose to form pyruvate and small amounts of ATP. This is your anaerobic glycolysis or anaerobic metabolism. And then we have slow glycolysis, where we see that pyruvate can be shuttled to the mitochondria. And so our second main metabolic pathway is the citric acid cycle, which occurs in the mitochondria. It completely oxidizes the products of glycolysis to produce large amounts of ATP through the electron transport chain. So these pathways represent the core energy systems that cells use to access stored energy in carbohydrates. And it just depends on the intensity and duration of the exercise, whether or not we have enough available oxygen to primarily utilize our aerobic metabolism or our anaerobic metabolism. Now, although glucose is the main carbohydrate, the main player, if you will, cells can also use fructose and galactose to produce ATP. These monosaccharides are first going to be converted into intermediates of glucose metabolism, primarily in the liver, before being used for energy. So while the body prefers glucose, it has mechanisms to handle other dietary sugars as well. All right, welcome back to part 2 of the lecture. Today we will go ahead and dive into glycolysis first. So first of all, glycolysis is the first phase of glucose catabolism, meaning that it's when our body begins to extract energy from the glucose molecules that are circulating. Now, it's an anaerobic pathway, meaning that it doesn't require oxygen, and it's going to take place entirely in the cytoplasm of the cell. Now, the goal of glycolysis is to break down the one 6-carbon glucose molecule into two 3-carbon pyruvate molecules. And these pyruvate molecules can then enter further metabolic pathways for additional ATP production. So when we take a look at the 10-step process of glycolysis, we see that the process actually begins with an investment phase where the cell must first spend energy to get the pathway started. So step one, we actually see that one ATP molecule is used to phosphorylate glucose, forming glucose 6 phosphate. And this step is crucial because the attached phosphate group, which you can see right here, that attached phosphate group traps glucose inside the cells so that it can't diffuse back out. And this glucose can come from three possible sources, right? You have your dietary carbohydrates. So these are the ones absorbed from the bloodstream. You have glycogen breakdown or small intestine into the bloodstream. You have your glycogen breakdown in the liver, which releases glucose into circulation for use by other tissues. And then you also have the glycogen that's stored within the working muscle cells, which can be broken down locally to glucose 6 phosphate. Now, only the liver has a specialized enzyme called phosphatase, which can remove the phosphate group in order to release the free glucose back into the blood. Other tissues can't do this. So this is why you can break down liver glycogen into glucose and allow it to flow to other tissues. Every other tissue, once we have converted the glucose into glucose 6 phosphate, we can't convert it back, right? We don't want it to diffuse back into the bloodstream. Now, after glucose is phosphorylated again for a second time, and this second time will form fructose 1,6 biphosphate. This molecule is going to split into 2 3-carbon fragments, which you can see right here. And so these fragments then proceed through a series of reactions to generate energy, or ATP. And what we see is that we get a total of 4 ATP molecules that are formed through this substrate phosphorylation. So you have 2 over here from this 3-carbon molecule, and then 2 over here from this 3-carbon molecule. Now we also have 2 NADH molecules that are formed during this process as well. Now, those NADH molecules can potentially shuttle the electrons through the electron transport chain if, say, exercise intensity is low enough or we're resting, or it can transport hydrogen ions to the pyruvate molecules to form lactate. It just depends on how intense we are with our exercise. So in this stage, we won't be too concerned with these NADH molecules. We'll get back to those 4 ATP molecules that were produced. And even though we produced 4 ATP, you still have to remember that we utilized 2 ATP at the beginning of this whole process. So the net gain is only 2 ATP, right? So we used 2 to make 4, so we only gain 2 extra ATP. Now, if glycolysis begins from free glucose, the cell invests 2 ATP like we just discussed, which means the net total would be 2 ATP. But if it begins from stored glycogen, we only need 1 ATP to be invested because that first phosphorylation step is bypassed because we are already storing glycogen in the muscle. And as a result, since we only use 1 invested or 1 ATP to produce these 4 ATP, we end up with a net total of 3 ATP per glycogen molecule. So for glucose, you are going to get 2 ATP during this process, whereas a molecule of glycogen, you will get 3 ATP through this process. And then the end stage of glycolysis will be pyruvate, which these 3 carbon molecules can then either be shuttled to the mitochondria to undergo the Krebs cycle and produce more ATP, or they can be converted to lactate. So again, it just really depends on the intensity of whatever physical activity that you are performing. All right, so as we just saw on the last slide, when we looked at glycolysis, you have that 1 molecule of glucose that is split into 2 molecules of pyruvate. And from here, pyruvate can go 1 of 2 paths depending on oxygen availability and the cell's energy demand, which in large part is based off of, say, exercise or physical activity intensity, like I said on the last slide. Now, pyruvate can be converted to lactate in the cytoplasm, or when oxygen is available, it can enter the mitochondria for complete oxidation, and that is what we see here, right? So we have the end product of glycolysis, pyruvate, and in this scenario, or on this slide, we will look at what happens to pyruvate whenever we are going to shuttle it out to the mitochondria or into the mitochondria. So under aerobic conditions, when plenty of oxygen is present, pyruvate moves from the cytoplasm, which is where glycolysis occurs, into the mitochondria, the powerhouse of the cell. So inside the mitochondria, pyruvate is converted into a new molecule called acetyl-CoA. And this conversion process is going to link glycolysis in the cytoplasm to the Krebs cycle in the mitochondria. And so the reaction is catalyzed by a large enzyme complex called the pyruvate dehydrogenase complex. And during this step, pyruvate, again, that 3 carbon molecule is oxidized, meaning it loses 1 carbon atom as carbon dioxide, or in the form of carbon dioxide, and the 2 remaining carbon fragments attach to coenzyme A, forming acetyl-CoA. And at the same time, you have NAD plus molecules that accept electrons and hydrogen that form your NADH molecule, which will later carry energy to the electron transport chain, which is going to contribute to the total amount of energy that we can accumulate or produce from this pyruvate molecule. Now, the coenzyme TPP, which is derived from vitamin B1, is also required for this reaction to occur. Now, when pyruvate is shuttled into the mitochondria and converted to acetyl-CoA, it can enter the Krebs cycle for further oxidation and a much greater ATP yield. However, because the process involves multiple enzymatic steps, the rate of ATP production is slower than anaerobic glycolysis, and this process is slower but more efficient, and is sometimes referred to as aerobic glycolysis or slow glycolysis. And so we see this will be the primary way we produce ATP during low to moderate intensity exercise. And it's the anaerobic glycolysis or fast glycolysis that we primarily utilize in high intensity activity. And this is where the pyruvate would just be converted to lactate to support anaerobic glycolysis because the energy demand is so fast. So if we go all the way back up to the start of glycolysis, right? So we have our beginning, which is one single molecule of glucose that completes glycolysis, gives us two pyruvate molecules, and each pyruvate will yield one acetyl-CoA and one NADH molecule and one carbon dioxide unit. Now, because this is all stemming from one glucose molecule, we see that we get two acetyl-CoA, two NADH, and two carbon dioxide total, right? Because you had that one glucose that split into two pyruvate. One pyruvate gives you one acetyl-CoA, one NADH, one carbon dioxide molecule, and then you have your second pyruvate that will yield these same molecules. So those acetyl-CoA molecules will then enter the Krebs cycle or the citric acid cycle to continue aerobic metabolism. All right, now taking a step back and looking at the theta pyruvate under anaerobic conditions, or when oxygen is limited, we see that when oxygen availability drops, like during high intensity exercise, the mitochondria can't process all of the pyruvate being produced by glycolysis. Instead of entering the mitochondria, pyruvate is converted to lactic acid for a split second and then quickly dissociates into lactate and hydrogen ions. Just so that you're aware, lactic acid can't accumulate in the body. It quickly disassociates almost immediately. We see that for lactic acid to accumulate in the body, you would have to have a pH below 6, which means you're probably dead at that point, because the survival range for humans, your normal average is about 7.4. Anything down to 7.8 is our survival range, but you can still see significant adverse effects even within that range. And so this conversion from glycolysis, from pyruvate into lactate is going to allow glycolysis to continue rapidly, even without oxygen. Now, the key reason this process occurs is the need to regenerate your NAD plus molecules. So remember, glycolysis relies on your NAD plus molecules to accept electrons, and without it, ATP production would stop entirely. And so the enzyme lactate dehydrogenase facilitates this conversion, turning pyruvate into lactate while oxidizing NADH back to NAD+. So we are taking those hydrogen ions from our electron transport molecules and donating them to the pyruvate molecules so that we can reutilize our NAD plus molecules, and you have your pyruvate that is then converted to lactate. And this rapid regeneration of NAD plus enables glycolysis to keep producing ATP, and we are able to produce ATP at a much faster rate this way, but it's a less efficient method than aerobic metabolism. Now, once lactate is formed, it will leave the muscle cell and enter the bloodstream. The liver then removes lactate and converts it back into glucose through a process referred to as the Cori cycle. And this newly synthesized glucose molecule can either be released back into the bloodstream so that other tissues may utilize it, or it could be stored as glycogen in the liver. However, this process is energy expensive, so it actually requires 6 ATP to remake that one glucose molecule. So it's not an efficient process that could be sustained indefinitely. Now, when we look at, say, our muscle fiber types, well, let's just cut it there and say your type 2 fast twitch muscle fibers will contain higher glycogen concentrations than your slow twitch. We won't go into muscle fiber types since this is nutrition. All right, so here you can kind of see the points that we were talking about illustrated. So inside the muscle or the muscle cell, we have glucose, which is broken down through glycolysis into pyruvate. Without oxygen, pyruvate accepts hydrogen ions and is eventually converted to lactate. So you see pyruvate accepts hydrogen ions, becomes lactic acid, loses some hydrogen ions, and results in lactate. And this reaction prevents hydrogen ion accumulation from shutting down glycolysis. So hydrogen ion accumulation would decrease the acidity within the muscle cell, and as a result, we would see fatigue set in and a reduction in ATP. But instead, what we see is lactate acts almost as a buffer, meaning that ATP can still be made fairly quickly. And this process without oxygen, of pyruvate being converted into lactate, is done via the enzyme lactate dehydrogenase. Now, once lactate is formed, it diffuses out of the muscle cell and into the bloodstream, and from here it can travel to different parts of the body and serve multiple metabolic purposes. About half of the lactate will stay local and be utilized either within the same cell or by nearby oxidative fibers like type 1 fibers. The other 50% enters circulation and can be used by other tissues or the liver. Now, this constant movement between tissues is called the lactate shuttle, and we see lactate isn't just a waste product, but it's also a metabolic intermediary. So it's transferring carbon and hydrogen between cells. Now, there are two main types of shuttling. You have cell-to-cell shuttling, so lactate moving from your glycolytic type 2 fibers to your more oxidative type 1 fibers, where it's converted back into pyruvate and oxidized for energy. And then you also have your intracellular shuttling within the same cell, right? So lactate produced in the cytosol can be transferred into the mitochondria, where mitochondrial lactate dehydrogenase converts it back to pyruvate for oxidation. And these mechanisms demonstrate the close integration between anaerobic and aerobic energy systems. Now, lactate that enters the bloodstream can also be taken up by the liver through a process called the Cori cycle. We see that the liver converts lactate back to glucose. And so this newly formed glucose can either be re-released into the blood, like we said, or stored as glycogen for the liver if, say, no immediate energy is needed. Now, this recycling of lactate is going to be vital during prolonged exercise, but again, like we said, it's energy costly because it requires 6 ATP per molecule of glucose produced. Now, once pyruvate is converted to acetyl-CoA, that acetyl-CoA enters the citric acid cycle. And so this is the second major phase of aerobic glucose metabolism following glycolysis. So remember, you had glycolysis wrapping up with pyruvate. And then once we decided to shuttle the pyruvate to the mitochondria, we converted it to acetyl-CoA. And now we see acetyl-CoA will enter the citric acid cycle, or the Krebs cycle. Now, the citric acid cycle occurs inside the mitochondria, and each acetyl-CoA molecule, remember, which is a 2-carbon molecule, combines with oxaloacetate, which is a 4-carbon molecule, to form a 6-carbon citrate molecule. Now, through a series of 8 enzymatic steps, that citrate molecule is gradually broken down back into oxaloacetate. And during this process, we produce carbon dioxide, which we exhale, hydrogen ions, and high energy electrons, which we can shuttle to the electron transport chain. And this will actually be where we see the bulk of our energy comes from. And then a small amount of ATP is produced. So only 2 ATP molecules come from this substrate phosphorylation. Now, the real energy payoff from this cycle isn't the direct ATP that is created. Instead, it's those hydrogens and electrons that are captured by NAD and FAD molecules. Again, the coenzymes are reduced into NADH and FADH2 molecules, which transport those high energy electrons to the electron transport chain, where the majority of ATP is made. So, in short, the Krebs cycle extracts energy from acetyl-CoA, but more importantly, we have the reduction of our NAD and FAD, our electron transport molecules or carriers that deliver the energy to the next stage. So because one glucose molecule forms 2 pyruvates, which will lead to 2 acetyl-CoA molecules being made, the cycle turns through that one glucose molecule twice, right? So one acetyl-CoA goes through the citric acid cycle, then another acetyl-CoA will enter the Krebs cycle. And each turn is going to produce just 1 ATP, so you get the total of 2. But you get 3 NADH molecules and 1 FADH molecule for each. So when we take a look at the total for one glucose molecule, which is 2 acetyl-CoAs, we get 6 NADH molecules produced, 2 FADH2 molecules produced. And your 2-ATP. So now you'll see that the NADH and FADH molecules that were produced during this process now move to the electron transport chain to undergo oxidative phosphorylation. And this is where the oxygen's role becomes essential and why we have to have a significant amount of it for aerobic metabolism. And it's because it serves as the final electron receptor, combining with hydrogen to form water. And then the flow of electrons through the electron transport chain powers the production of the majority of ATP in aerobic metabolism, which is going to fluctuate depending on what resource you're referring to. But here we are going to say that for each NADH molecule, we are going to produce 2.5 ATP. And for each FADH2 molecule, we will produce 1.5 ATP. And so that'll give us a gross yield of 34 ATP produced from one single glucose molecule. All right, so here you can see the citric acid cycle illustrated. Once pyruvate enters the mitochondria under aerobic conditions, it's converted into acetyl-CoA, which then enters the citric acid cycle. And this cycle represents the central hub of aerobic metabolism, where carbon, hydrogen, and oxygen molecules from our macronutrients are processed to generate energy. And so on this diagram, you can follow the process in a circular flow, showing how acetyl-CoA enters, it's oxidized, and eventually regenerates the starting molecule oxaloacetate. So when we look at step 1, we are forming citrate. So the cycle begins when acetyl-CoA, a 2-carbon molecule, combines with oxaloacetate, which is a 4-carbon molecule, to form a 6-carbon molecule referred to as citrate. Now, this reaction is catalyzed by citrate synthase, which is one of the key regulatory enzymes in metabolism. Now, this step commits the acetyl group to oxidation. So once it enters the cycle, it can't be converted back to pyruvate or glucose. Now, as citrate moves through the next series of reactions, we see 2 carbon dioxide molecules are released, and these will ultimately be exhaled through respiration. And also during these steps, we have several NAD molecules that are reduced to NADH, meaning they've picked up high energy electrons and hydrogen ions. And this kind of represents an energy harvest, right? So we aren't producing ATP yet, but these electrons will later drive ATP production in the electron transport chain. Now, in one specific step, so when succinyl-CoA is converted to succinate, a small amount of ATP, well, I guess technically GTP, is produced and is then converted to ATP. But this ATP is essentially formed directly through the citric acid cycle, and this is known as the substrate level phosphorylation. So although the cycle's main purpose is to provide potential energy that will be directed towards the electron transport chain or produced at the electron transport chain, it does also yield a small amount of ATP. So the remaining steps of the cycle convert succinate back to oxaloacetate, which allows the cycle to continue as long as acetyl-CoA and oxygen are available. So each full rotation of the cycle processes one acetyl-CoA molecule, meaning that from one glucose molecule, we get 2 full turns through the citric acid cycle. All right, and then finally, we've arrived to the last and final phase of aerobic metabolism, the electron transport chain, where the majority of ATP is going to be produced or generated. So you can think of this as the energy payoff stage. So all those NADH, FADH2 molecules that were produced during glycolysis and the citric acid cycle are now going to deliver their electrons to make ATP. And so this process takes place on the inner membrane of the mitochondria, and the electron transport chain is a linked series of enzymes and cytochromes, which are specialized proteins that pass electrons from one molecule to another in a stepwise fashion. Now, each transfer releases a small amount of energy, which is used to pump hydrogen ions across the inner mitochondrial membrane. And this creates a proton gradient. And so those hydrogen ions then flow back through an enzyme called ATP synthase, which uses that gradient to phosphorylate ADP into ATP. And this coupling of electron flow and ATP formation is referred to as oxidative phosphorylation. Oxidative because this requires oxygen, and phosphorylation because a phosphate group is being added back to ADP. And so the final electron acceptor in this process is oxygen. Like we said, it's required for this process to occur. And the enzyme cytochrome C helps facilitate the transfer of electrons and hydrogen ions to oxygen, forming metabolic water, or H2O, as a byproduct. And this is, again, why oxygen is essential for aerobic metabolism. Without it, the electron transport chain stops, it gets backed up, and so the NADH, FADH2 molecules can't unload their electrons, and ATP production comes to a halt. And so when we look at the total amount of ATP from the entire process, from the start of breaking down the one glucose molecule to the end of the electron transport chain, we get approximately 32 molecules of ATP. And so when we break this down, we see we have the 2 ATP as the net yield from glycolysis earlier in our discussion. And we also had those 2 NADH molecules that were produced. And each one of those NADH molecules yields approximately 2.5 ATP. So we have that additional 5 ATP. Now, we also have ATP that will be produced when pyruvate is converted to acetyl-CoA, or I shouldn't say produced at that moment, but we have NADH molecules that are produced, which will yield another 5 ATP because we have 2 pyruvate molecules that will be converted to acetyl-CoA. And then you have your citric acid cycle, which we run through twice because we have 2 pyruvates, 2 acetyl-CoAs. And so as a result, each citric acid cycle produces 3 NADH, one FADH2, and one ATP. And so when we look at how much was produced through the one glucose molecule, we come out with 6 NADH molecules, which will have produced 15 ATP through the electron transport chain. We have 2 FADH2 molecules that are going to produce 3 ATP once they deliver their electrons to the electron transport chain. And then you have 2 ATP that was directly produced through the citric acid cycle. And so from there, you had 20 total ATP. So when you add everything up, you had the 7 ATP from glycolysis, the 2 net yield plus the 5 from the NADH molecules. You had 5 from the conversion of pyruvate to acetyl-CoA. And then you had your 20 from the citric acid cycle and the electron transport chain. And as a result, you come out with a total of 32 ATP that were produced from one glucose molecule through aerobic metabolism. And so this slide just ties everything together. So the complete breakdown of glucose for energy. We've talked through each phase individually, so glycolysis, the citric acid cycle, and the electron transport chain. And this diagram just connects all of these phases together. So you can think of this summary as like a roadmap of aerobic energy metabolism, where we have approximately 32 ATP that will be generated at the end. So again, running through phase 1, you have glycolysis, which begins in the cytoplasm outside of the mitochondria. We have that one glucose molecule that's a 6-carbon molecule that's broken down into two 3-carbon pyruvate molecules. And then again, glycolysis is anaerobic. It doesn't require oxygen, and it's going to give us a net total of 2 ATP directly. And then it produces those 2 NADH molecules, which can be shuttled to the mitochondria later and converted to another 5 ATP in the electron transport chain. And then phase 2, you have each pyruvate molecule that enters the mitochondria, where it's converted into acetyl-CoA, producing 1 NADH per pyruvate. So you get 2 NADH per glucose just from converting your pyruvates to acetyl-CoA. That's, again, a 5 extra ATP from those 2 NADH molecules, again, being generated at the electron transport chain. And then you have the acetyl-CoA that will enter the citric acid cycle, or the Krebs cycle, where it's further oxidized. And for each acetyl-CoA molecule, we get 3 NADH, which comes out to 7.5 ATP, 1 FADH2, which comes out to 1.5 ATP, and 1 ATP via the substrate level phosphorylation. And again, because we have 2 pyruvates and thus 2 acetyl-CoAs, we double that number and we end up with about 20 ATP total from the Krebs cycle. And then again, looking at the electron transport chain, you have all of those NADH, FADH molecules that carry their high energy electrons into the electron transport chain. And as those electrons move through the cytochromes, their energy is used to pump protons and drive ATP synthase, which phosphorylates ADP back into ATP. And at the end of the chain, oxygen acts as the final electron acceptor, combining with hydrogen to form metabolic water, which again is shown at the bottom of the screen right here. And so when we total all ATP production up from phase 1 to the end, phase 3, we get approximately 32 ATP.